coral reef expert, and he's also a professor emeritus at Tel Aviv University, where he's been a faculty member for over 40 years. He's published several books on coral reefs and authored over 240 publications in leading international journals, including 7 papers in Nature and Science. His seminal publications on biodiversity, coral reproduction, coral bleaching, and conservation have become classics in the ecological literature. He's also chaired numerous high-caliber scientific and governmental committees for Israel and was founding editor of the journal International Journal Coral Reefs. And in addition to all of his scientific work, he's also been a really big advocate for coral reef health in Israel and around the world. So, without further ado, I'd like to introduce Dr. Yossi Loya.
No? Oh, right now. Okay, great. So, thank you very much, Natasha. I'm really excited of being here because Stony Brook has been a home to me during my graduate studies with Larry Slobodkin, the late Larry Slobodkin. I'm really happy to see here Tommy, his wife, and I'm too excited to see you. The Slobotkins have been family to me, and thank you very much for coming. So what I'd like to do now is tell you about reproduction in corals. I've been working on reproduction of corals a long time. And towards the end of my career, I think I came up to the main story that excited me in terms of what is happening in biology of corals. So I've been working on different species. What you see here are fungids, which are the main subject for today, and I'll talk about reproductive strategies in fungids, which are very, very different than many other corals. I've been doing this study in Eilat, which is in Israel, in Aqaba, which is in Jordan, and mainly in Japan, in Okinawa. It's a very large project, as you will see, but here are some of my students over the years working about this thing, and just I wanted to make sure that you'll see some of the faces that worked with me throughout the years. So the outline of my talk, I'll give a brief review on the sexual reproduction in stony corals, the reproductive traits of fungid corals, these are the mushroom corals, and especially on sex change, which is the main subject today, senescence in reproductive effort, and possible cues for sex change. So before I start, I'd like just to make sure on some terminology that not everybody is probably familiar with. And although the majority of animals exhibit stable sexuality, it means that they stay the same sex throughout their lives and reproduce as gonocores, which means that they have separate sexes. We have males and females. Many reproduce as hermaphrodites. And hermaphrodites, in terms of terminology, again, we have what is called simultaneous hermaphrodites. They function in both as both sexes consecutively or concurrently, or we have sequential hermaphrodites, which are the true sex changers. Now, another term that we have to know that most animals changing sex change will occur only once in an individual's lifetime, either from male to female, which we call protandry, or vice versa, which we call protogeny. And this is what we know today, and this is a matter of fact of summation of all the information that we know about coral reproduction. I must tell you that until the beginning of the 70s, almost nothing was known on coral reproduction. All what we knew is what Libby Hyman's book told us, or some other textbooks. And the general idea was that most corals are hermaphrodites, as you can see here, and they are also brooders. in the beginning of the 70s, all what we knew, most corals are brooders. They are have internal fertilization, that is, sperm is fertilizing eggs which are within the coral, and they produce planarly into the environment. That was certainly not the story, as you will see later on, when, as a matter of fact, most of the corals are hermaphrodites. Okay, in a matter of fact, we divide them into two sizes, into two categories, hermaphrodites and gonocores, where 70%, 75% of the corals are hermaphrodites and 25% are gonocores. Most of the hermaphrodites are simultaneous hermaphrodites, okay? You'll see most of the species, as a matter of fact, are simultaneous hermaphrodites, and the minority brood planarly larvae that we thought that is the rule. Now, if we look at the gonocores, that is where sexes are separate, we see that most of them spawn gametes. In other words, they have separate sexes. They spread their gametes into the water column where fertilization takes place. I will talk today also mainly of this part of the graph, the brooders, which is a story that hasn't been told yet, hasn't been published yet, and I find it of much interest. So just to give you a little bit of background, this is a colony, Stylophora pistillata, which is the most abundant coral in the Red Sea, and the most important one. It's the most important one since it's also supported my graduate students for the last 40 years in terms of grant proposals and so on. There are about 4 PhDs on this coral and several masters. But what you see here is the colony. These are the polyps, as you can see, and we'll go, is one polyp. We'll go very fast through what we knew in the beginning of the 70s. And what you can see here is an histological study where we have in the same polyp, you have the female gonad and the male gonad side by side. This is the nucleus, the nucleolus. The fertilization is in the water column. And here you can see a planular larvae in cross-section starting to develop. And this is the mature planulae. This is being shed to the environment. And here you can see the planular larvae that is elongated about 1 centimeter or so. It changes its shape to a pear-like and changes its feathery shape until it settles down. And here you can see an isograph of 2 of these planulae, as a matter of fact, touching each other and creating a coral, growing up a primary polyp, a young colony, to a big colony. This, more or less, this is a colony of about 10 or 12 years. So that's one of the fastest corals that grows, and that life cycle is very clear. This is what we have known in the beginning of the 70s. Now, the big story came later on from the Great Barrier Reef, where a group of graduate students have been studying reproduction of corals. And they found this amazing story in terms of coral biology. And that is, I'm sure many of you know the story, of course, that 130 different species of corals are reproducing in one night only. So it's a big orgy of corals that are spreading their sperm and eggs into the environment in one night only. This is 3 or 4 nights after the full moon of September or October. And you can ask lots of questions about it, why and what are the cues and so on and so forth. But that's not what I want to tell you. I want to tell you what happens in the Red Sea, which is exactly the opposite. So here you can see the egg and sperm bundles of these hermaphrodites. Here is a bundle of eggs and sperm. And here you can see that just before shedding into the environment, okay, you see all these eggs and sperm being shed. This is a minute before being shed out, and this is immediately later on. And this is the result the next morning. So all what you see this red about, sometimes when you fly over a reef, you see miles of this red stuff. which is a night after this reproduction. All these eggs were not fertilized, and they are right floating on the surface. So now, this was a big story for coral biologists, and it made a big different change because it was totally different than what we knew until then. It's not brooders, it's broadcasting. And the story was even more interesting because what we found in the Red Sea is a totally different story. The same species, exactly as the Great Barrier Reef, reproduce in different times or different months or different moon phases. So what you see here are the most abundant corals in the Red Sea. As a matter of fact, the list is more. We have about 26 of them that now we know. And what you have here is the timing of reproduction, where the black bars indicate when they're reproducing according to the moon phases. You see the first species, this Stylophora, that reproduces like almost every month during wintertime. Note that there is no overlap in this, the reproduction times of these species. So I'll just go very fast. And the amazing story was that there was temporal reproductive isolation. One species starts to reproduce on Sunday, ends up on Tuesday, then the other one picks up, and the other one picks up. So they divide the time of reproduction throughout the months of different moon phases or different months throughout the summer. This, of course, makes much more sense. It's easier to explain in terms of reproductive success. But the big story of the Great Barrier Reef still is not totally solved, because look at that. It takes several months for a female gonad to develop. It takes more energy to build a female gonad than a male gonad. That's where the female starts, for example, in this species, starts developing. Then the male starts developing. They all end up at the same time, and they are synchronously shedding their eggs and sperm to the environment. Now think about 130 different species that have different times of development of their gonads, yet all of them exactly at the same time are fully developed and shed their gametes into the environment. So this is a big wonder. We'll not go into it because that's not my story today, but just to show you that corals have different strategies of reproduction. And what I've showed you in the big review of what we know about corals is, as a matter of fact, the secret why they're so successful in evolution. They're so successful because they have this high plasticity of methodology in reproduction. And as you can see here, they are all reproducing in different times. As I said, I worked a lot in Japan, and I was lucky to be there. As a matter of fact, since 1995, I spent many summers in Okinawa, and I did my studies of mainly community structure and species diversity. So I had a lot of data in many localities around Okinawa as to the community structure of corals. Species lists, abundance, and so on and so forth. Now, in 1998, which was the hottest year to be recorded until then, there was this disaster of coral bleaching all over in many reefs. What happened in Okinawa, that there was a total catastrophe in a way, and you can see it in the following graph. If we'll measure the percent or number per square meter of living cover of soft corals and stony corals, of stony corals alone, soft corals, number of colonies, number of species, all these per transect, then this is the picture in 1997, a year prior to this bleaching event. So there's pretty high cover of both stony corals and soft corals. We're talking on cover of about 30–40% in stony corals and soft corals, which is pretty high. It's pretty good. Now note what happened a year after 1997. This is 1999, after this bleaching event. You see that there is a disaster in terms of number. What was the cover that looked like at that time in the stony corals, in the soft corals, and in the number of species? As a matter of fact, until today, some of the species are locally extinct. They're not there anymore. And what we put then together, I mean, you can see here the percentages of decrease, 85%, 61% of the number of species, and so on. This just tells you the whole story. I mean, the whole reef became white. All these corals just died fairly quickly after this bleaching event, as you can see it here. The soft corals were decaying, so the smell of the decay, you could feel it all over the reef. It was a terrible sight. And that was the result. The algae took over, and the reef was dead. The reef is dead to large parts of Okinawa until today, as a matter of fact, although there is some recruitment coming from farther islands. We published then a paper which was coral bleaching, the winners and the losers. After every disaster, you have some winners and you have some losers. What happened was that some corals that were very low in abundance took over, and there was a community shift, that some of the corals were winners, others, as I said, simply are not there anymore. They are locally extinct. These fungids, the mushroom corals, came into the picture of this story. By a chance, we dived in a particular reef that we haven't been there before, and what I see is thousands and thousands of these mushroom corals, and they haven't been touched. I mean, they went through the bleaching event, they're okay, nothing happened, so you ask why. But my interest was more in reproduction. Here you can see some photos of these species that we studied with time. All of them are fungid, these mushroom corals. I'll tell you later on the story of this species. More and more, all of them are one polyp. In other words, in contrast to what you've seen before, colonies, a whole colony is composed of many polyps. Here, it's only one polyp, and it's also not attached to the substrate as all other corals. In other words, a great advantage of working with them is that you can take them out of the reef, bring them to the lab, take them individually, and follow their growth, follow their whatever you like to do with them. So this is what I've been doing both in Okinawa, Eilat, and Aqaba. And I'll tell you the main story coming out of Okinawa. And this is Okinawa, okay, Okinawa Island. Here is the reef that I've been working on. This is a bridge that connects between mainland Okinawa and the island of Sesoko. And I'm showing you this photo to illustrate what happens during bad years. So sedimentation is one of the big problems there. After a typhoon, there is a lot of sediment which runs into the reef. All this red silt runs onto the reef. You can see it here. And when you're in these areas where you have all these fungi, you can hardly see from 2 or 3 meters away. It's really like a rain of silt coming up from upwards. That's the way the bay looks like after such a typhoon, where it's all covered by silt. And this is the bridge that you have seen before in good times, and this is the same bridge after such drainage and after such a typhoon, where you can see all this silt coming to the reef. Now, this has a lot of effect, of course, but these fungids are doing fine. They are thriving even in such an environment, and one solution was that they simply can clean themselves by secreting mucus, by tentacle movement, and some other ways. So this is what we term fungiland. It's this particular spot, there were thousands of fungus there, and you can see it here. Okay? This is when the whole story started. There were about 12 different species there, and I wanted to study their biology, their reproduction. So within half an hour, you can collect 300 of them as if you didn't touch them. You didn't touch the area. There are so many there. So there was no problem as far as that is concerned. You can see bringing them back to the lab. And as I said, the good thing about working with them is that you can take the whole coral, which is one polyp, bring it to the lab and tag it, and nothing happens. They're very strong. And then you do what you do with them, and then you bring them back to the reef, and you can do this over and over again. So this is the experiment that we put. We put each one of this coral in a separate aquarium or a separate container. As you can see, each one is tagged with a number. And to make a long story short, we didn't know at that time nothing about their reproduction. And one night, after looking at them, measuring them, we see that they shed eggs and sperm into the separate aquaria where they are. So we knew that this guy, number 27, is a male, and this is a female. This one is a male, and this one is a female. We knew individually by number which one, what is the sex, and all the history of growth, and you name it. So here is the system of this aquaria with running seawater, and again, the experiment, and then the story starts 5 to 7 nights after the full moon of June, July, and August. We do know that full moon is the loving time in many invertebrates, but different invertebrates divide the timing after or prior to the full moon as to when they reproduce. These corals are doing it 5 to 7 nights after the full moon. So if you come to Okinawa in July or August, 5 days after full moon, 10 o'clock at night, watch the aquaria, you see them spawning. The timing is amazingly exact. This biological clock is simply amazing. Not only that, but the same individual will reproduce year after year after year exactly at the same time. And you can look at the watch and see that. So this is one thing which is amazing about their biological clock. Also, note that it's very easy to say what is a male. Here you see male, and what is a female. I think I have a better, no. There is no way to make a mistake as to what is a male and what is a female, because the sperm is a whole cloud, white cloud. You don't see the coral below. I hope I have another slide to show that. But here is a female which is shedding eggs, as you can see it here, okay? And usually it's from openings from the mouth, some openings in the polyp. And this is a male shedding its sperm. It goes like a clockwise every 5 minutes. There goes like a blow of sperm, and then 5 minutes rest, and then again. Here you can see this same thing, like the sperm ejection throughout the mouth, and you can see the cloud of sperm coming out here. This was from last summer, and I was very excited to see this about this species. So it's not only for one mouth, but you see that they have multiple mouths. And then you can see all these spurts of sperm coming out from this particular polyp. Remember, it's one polyp only. And again, they're doing it like you see it in 1 minute, you wait for 3 minutes, then it goes again. You wait for 3 minutes, and then it goes again. And here's just to show you how there is no problem to differentiate between males and females, so there was no mistake in saying who is a male and who is a female. Now the surprise was that after doing this for 2 or 3 years, and they're doing it at night, I'm sitting in the lab and watching the numbers, and I see that number 27 is a male, and number 36 is a female. And then a year later, this one that was a male is a female now. And this one that was a female is a male now. So I said, there is no way, it's a mistake. So I'm looking and there's another number and another number and another number. So it strikes me we have a sex change here. So we never knew anything about that in corals, and indeed that was a big, big surprise. But even the surprise was even more that not only that they're changing sex from male to a female or vice, they're also doing it bidirectional and multiple times. So this study is already going on for a few years. So we do know that a particular individual started as a male, changed into a female, back to a male, and then you ask why and what are the cues and so on that we will see in a minute. So the story of sex change in fish is pretty known for many, many years. So you may ask, what's the big deal about sex change? The big deal about sex change in corals is that they do not have behavior. Like in fish, what you have here is a male which has a harem of females, and being a male, he shows himself how strong he is and so on, so he's the first one to be preyed upon. And once he's being preyed upon, female alpha takes over and she becomes a male within 2 weeks. So this is fairly known from different fish, and they have different strategies. But again, in corals, we didn't know that. And how come? Why are they doing it? What is the advantages of doing it? So this is a graph showing just a allometric relationship between the size range and at sex change of these two different species. We have here this elongated form, Ctenactis echinata, and this rounded form, which is this graph, and this is this graph. The blue dots are females, then the red is males. These are individuals which are not in reproduction. Every dot here is an individual coral. So what we see here, that in very early age, as you would expect, most of them are males or are not in reproduction. In intermediate ages, this is the time where they change sex. When they're coming older, in this case, they're all remaining females. It's a different story here, although again, starting, they're not in reproduction, or either they are males. Here, they change sex. And what is interesting here, that all of these guys, the very old guys, are not reproducing at all. So once they're becoming old, they do not reproduce. So maybe what we have here is senescence. And the next thing to study was, are they senescing? So remember, we're talking on one polyp. One polyp is getting born, it grows up, and it senesces. It's not like a colony that philosophically coral colonies do not die. They have indefinite life, okay? This is just one polyp. So we wanted to study also this story of senescence. I'll skip that just to show you that these are all species that we studied with their different reproductive strategies. I will not go into this, but I'd like to concentrate now on this particular species, which is a story that is developing now. In other words, we not only that we didn't publish it, it's incomplete, but it's fascinating in the sense that it's very different from all other fungi that you have seen before. I'll skip the histology, which is boring, tedious, but very important in this kind of study. And here you can see all the development from an unfertilized egg through a immature larvae, going up to settlement. This is a cross-section through a larvae with 6 septa and the first settlement of such a larvae. It's amazing to see in a petri dish when you see a planula larvae swimming in seawater right in front of us, settles down and secretes calcium carbonate, and there you get a coral under your eyes. And then within a minute, he or she decides that I don't like the place, and then they leave off with a stamp of calcium carbonate in until they find a place where they like, and that's where they sit for life. These are most of the corals, or almost all of them, except the fungids that are, in a way, free-living. And this is the planula larvae of this particular species. Now, if all other species that I've showed you before are hermaphrodites and are shedding sperm and eggs into the environment, this species is developing planulae. Again, for me, that worked all his life with reproduction in corals, and that was a big surprise to see a fungid which produces planula larvae. Never seen before, and if you're not experienced, you'll never see them. So here you see it after many, many trials and photos, but they look like so tiny, like the edge of a needle. They're white. You can't see them unless you're really experienced and know what you're looking. So this is the planula larvae. As you can see, it comes already with a zooxanthellae. These are the mesenteries, and all what it is, it's about 270 microns. So it was quite easy to count also the number of planulae which are produced by each of these specimens. And the way we do it, we put this in these containers. This is, by the way, in Aqaba. I had a seminar given there. So for us, Jordan, to work in Jordan, to work in Aqaba is a big thing. And as you can see, it's a very successful collaboration. We're very happy about it, although it's on and off. So what we do here, we watch each individual, how much eggs, how much sperm, or how many planulae they produce, and we count them. So the whole idea is to have and to study reproductive success, or in effect, if you like, fitness in this species. Most interesting, this particular species, as you can see, while in Japan, in Okinawa, produces planulae, the same species. In Aqaba, it's shedding eggs and sperm. Same species, and we've shown this genetically. Same species behaves totally different in two different geographical localities. We know some other examples about that, but of course it was quite surprising to see that. Now, this summarizes, as a matter of fact, this individual sex change in different corals, and here you can see the coral number, okay, individual number 71, 76, and so on and so forth. And note that all the very young ones, those who their diameter is very small, they're all changing from female to male, female to male, female to male. In other words, when they're very young, they're changing from female to male. This is also against our thinking as to on the energetics of this, because when you start as a female, you have to invest much more energy into building up a female gonad than into building up a male gonads. So how come you start as a female while all the others start as male and then they change into female? Here, the middle age are changing from males to females, and then the old guys from females to males. If anyone gives me a good explanation, I'd love to hear that. I even didn't have enough time to think about it. But it is definitely the other way from what we've known from all other species among the fungids. So we're talking here on protogynous sex changer. In other words, starts as a female and continues, as a matter of fact, as a male, and then may change over. This is the story of individuals in terms of their sex change. Each one of these bars is a particular individual. That is the weight. This is the diameter. Again, all these guys in the middle size change from a male to female. The very young ones and the very old ones change from female to male. I'll skip that and just tell you that out of 51 individuals that were in the sample, 35 changed sex, and in terms of percentage, then 25 of that changed from male to female, 75% changed from female to male. This may be explaining something as to what is happening here. I will skip that because we already say that. Now, the next question, as I mentioned before, since we're talking on individual polyps, not on a colony, so do they senesce? And do they senesce reproductively? We counted the number of planulae, we counted the eggs, we counted the sperm, and we knew their ages. You can detect the age of a fungid by their growth rings that you can see here. Okay? So note that this is a year of growth, another year of growth, another year of growth, and note that in the early days of their life, their growth rate is much faster than later on. This is about the same period of years, much faster. This is very nice, but this is not the only method that you can that rely on, because not all of them are showing such nice growth rings, and you definitely do not want to kill your animal just to know its age. So another way to do it is by doing a computerized tomography, and that you can see here, and this is only in Japan. It costs a lot of money, but it's free there. So we managed to get these growth rings doing CT, as you can see it here. And all what I can tell you is that we know exactly now the ages of the corals, not only based on that, but based on following them for years in terms of their growth rates. Different age groups have different growth rates, and you can devise a whole growth rate curve, so we know exactly their ages, which are pretty well correlated with their weight. So if we have here for example, in terms of fecundity and senescence, we count the total number of eggs of particular individuals. Each one of these points is a different individual. As you go to the right, you age. There are different big colonies. As I said before, when you're very young, the number of eggs that you're producing are small, until an optimal size where it's a maximal of production of eggs. And as you senesce, as you grow, the number of eggs that you're producing is much, much lower. So yes, there is reproductive senescence in corals in this particular species. If this is true, then what we expect is that these guys, the very young ones, will have long telomeres, while these guys that senesce have short telomeres. And that was the next step to find out, do they really senesce? Do their telomeres show senescence? So I will not go into this, but I'm sure you're all familiar with that. If this is a cell and this is the nucleus, these are the different chromosomes. The telomeres, of course, form the caps around the ends of the chromosome, and they help, as a matter of fact, to protect the chromosomes from degradation and fusion. And also, they shorten during cell division. In other words, and you know, these are the guys that got the Nobel Prize in 2009 in finding, as a matter of fact, the telomerase that restores the length of telomeres. Now, this is a well-known graph that the telomere length shortens in dividing cells as we age. So when we are aged 1, this is more or less the length of our telomeres. Okay, when we are 35, this is the length. When we're 65, it's around here. When you are my age, it's around here. So it goes down all the way. Clearly, there is correlation between the length of telomeres or the base pairs and age. We did find this in corals, in this particular coral, because we knew their ages. We could compare sperm of a given individual, which is somatic tissue. Unfortunately, and this is working with Japanese, convincing them, we have to have a larger sample size. We have to repeat that. We have to, very hard. So unfortunately, these two curves are still not significant to say that indeed there is shortening of telomeres in corals, but it will come. Do these species senesce reproductively? So this is the other species, the fungi that I told you before, that it's easy to work with because they produce planulae. It's easy to count the number of planulae each one of them is producing. And what you can see here is the same story. Here we have female fecundity, the number of planulae produced versus the diameter or the size of the coral. And you can see again very low when they're young. When they're in mid-size, they produce maximum. And as they age, you see that the number of planulae they produce is almost zero. So there is a certain size, an optimal size, when they are most fecund, which is around 70 millimeters. The same story is with the males, and also about this magic number or magic size is the maximal fecundity of the males. Putting them together, again, it's more or less the mid-size that they have their maximal fecundity, but the story again is that they do senesce reproductively as they age. This is summarizing this story of the reproductive senescence in these species. There is no significant difference in sex ratio when you take the whole population, as a matter of fact. But if you divide them from this 70 millimeter diameter, this magic number, there is a difference. While there is no difference in sex ratio between males and females, here there is a big difference when they age. It is 5-to-1 in terms of males versus females. But note again what you see here, the reds are the females, the blues are the males. They start as females. You tell me why. I have some thoughts about it, but it's not the end of it. So definitely reproductive aging occurs most probably due to individual senescence. So the question is now, why change sex? What are the advantages of changing sex? And how do we explain the repetitive sex change in fungi? This is the obvious question that we have to ask. So the obvious answer is to maximize reproductive success or your fitness. That is your genetic contribution to the next generation. Now this story has been long known in plants. And I think that Charnov, with his book in 1982, is the Bible of the theory of sex allocation. And he showed in his models also, which is termed the resource limitation model of sex allocation theory, which predicts that an increase in plant size and associated resource availability will maximize plant reproductive success or fitness through an individual increase in female expression. I know that this is not very clear right now, but it will be in a minute much clearer. To give you an example is this orchid. There are many orchids that if you grow them in bad conditions, little water, drought, no nutrients, they are males. If you put them in good conditions, light, appropriate nutrients, water and so on, it turns into females. So the whole idea is a female is more expensive than a male. You need much more energy to produce eggs and sperm or a female gonad than you need for a male gonad. And the whole thing is, as a matter of fact, an energetic expenditure story as to when you do what in terms of sex change. And there are other examples of plants that are doing it, but we are thinking in zoology or that we're inventing the wheel all the time, while plant ecologists have said it a long time ago in examples of plants. And this is well-known phenomenon in different plants. But corals, you know, the whole story of coral reef ecology started, in effect, took over in the 1970s. The first coral reef symposium was in 1969. There were only 18 papers then, 18 scientists. That was in India. Since then, every 4 years there was a International Coral Reef Symposium, and today you have 4,000 or 5,000 people in such. So that tells you about the explosion in information in coral reef ecology. Here I want to show you the size distribution of a particular species and individual. You see that these are small. Okay, this is the size what I termed small. This is intermediate size, and these are very large. And I'm saying this because in a minute you'll show you why size is important in that story. And just to show you what size means, so this coral, this fungid, is 95 years old, even older than me on this boat. 95 years old, and this is the fungia fungitis that you have seen before, which is a very large individual compared in this species. So you see that there are different sizes. Here now we'll go into sex allocation theory, which is the way in which organisms allocate resources to male and female function, and from that into the size advantage hypothesis that I'd like to show you here. So the SAH, or the size advantage hypothesis, predicts that sex change is favored when reproductive success or fitness increases more quickly with size or age in one sex than in the other, as you will see in a minute, and I know at this stage maybe it is not totally clear. But this will explain you what the hypothesis is. So if we plot reproductive value or fitness versus body size, and we have two curves of fitness, one for the male and one for the female, which is all theoretical. And note here that the female curve, or if the female fitness increases at a faster rate than the male, you see it is above the male, when they are young or small in size, and then they slow down as they age, that is the point, this particular point, where it is worthwhile for the female to change sex, because then it will go up. Okay, so if that female will not change sex here, then it may lose fitness in a way, it may lose reproductive success. So this is the point where it's worthwhile, again theoretically, to change sex, and that but as a matter of fact is one form of what is called protogynous sex change from female to male. That's what we've seen with the smaller fungia fungitis coral. Of course, the opposite is if the male fitness curve is above that of the female, it's exactly the opposite. Here is the turning point, and this is the point where a male should change into a female, and this is the protogynous sex change, or change from male into a female. In other words, it is more advantageous to this particular individual, which is a male, to change sex around here, about this size, turn into a female. You increase your fitness, you increase your reproductive value. These are the species that were found to follow this model, and this is the fungia fungitis, which is still developing story and incomplete. The next question is, what are the possible cues for sex change? Is it behavior? Well, it's not like fish where they change sex because of behavior, as you have seen in previous slides. So maybe it is energetic and environmental constraints, like we have seen the problem of sedimentation, light deficiency, breakage, or bleaching diseases, and so on. Maybe that is what a cue to change sex, or maybe there is a chemical cue or hormones. So we try to test that. The problem with corals is that they are not microbes. You have to wait a whole year to do any experiment that you do in reproduction. You have to wait a whole year for results. So we do an experiment, we do some changes, we bring them back to the water, then we come the next year, we take them out of the water, and see what have we got. At my age, it's a bit problematic in turning, running with time with these corals. It takes a whole year. And, well, to test the problem of the hypothesis that the disturbance may be a reason for sex change, I cut them into two. Okay, this is a female, large female, cut into two. I know at the point of doing this half is a female, this half is a female, but this is a back disturbance, okay? Now they need to heal, they need to regenerate. They have to turn energy into regeneration. That takes energy. So here's just to show you that it's not as bad as it sounds. They regenerate pretty fast. This is, you see here, the regeneration of this particular one. This is after 4 months. This is after 12 months. The interesting thing is if you take the rounded ones and you divide them into 4, within 3 years they regain their former shape, and they are bigger than the mother colony, the mother individual. So they regenerate very fast, and as a matter of fact, they regain their shape. So as I said, they're very easy and very good to work with. So the experiment to do then, to test if there is any chemical cues, in other words, maybe there are some hormones that are being secreted by individuals which are sitting on the reef close to each other. Saying, I'm a male, you're a female, let's check whether it's better for us to check to change sex, and thereby we will increase our fitness. So one can do things like that. I married them for life. Not for life, for a year. I'm sorry, for life they will kill me. Just for a year. Here you can see a small male and a large female which are tied together, and this is a matter of fact a waste for both of them, because a small I'm sorry. Yeah, right. The ultimate experiment, as a matter of fact, there are very, very many other variations here that can happen. And after a year or two of experiments, very quickly you realize that your sample size is not big enough, that biology is not exactly mathematics, and you have to have one experiment at a time with very large sample size. So in this case, when you have a small male and large female, this is the case where both of them are happy, both of them will not change, also thinking energetically, will not change the next year. But this is the ultimate experiment. Having a small female tied with a large male, it's a waste for both of them, because a large male can fertilize many females. What you would expect if theory is right and the size advantage hypothesis is right, that a year later on, this large male will become a large female, and this small female will become a small male. So think about it a little bit, and it will make sense to you. Anyway, this is the experiment, tying them together. You see a small and a large, and always the male is larger, the female is smaller, and this is, of course, after several years of knowing who is a male, who is a female, how did they change, you know the history of each individual, and then you can do that. And you marry them for a year, you tie them, you put them on different localities on the reef, far away from each other, far away from any other corals, so that if there is any hormonal cues between the two, it will happen between the two and not from other corals that are nearby. And here's the way it looks in the field. You see here different couples, okay? This experiment is going on now in Okinawa, in Eilat, and in Aqaba. So in all three places, we're waiting to see what will happen from this marriage, and if indeed something will come out in terms of hormonal signs. Yeah, this is the sign that I'd like to end up, and this is with a statement from Darwin in his book, The Descent of Man, 1874. And what he says there, that I formerly thought that when a tendency to produce the two sexes in equal numbers, that is 1-to-1 between a male and a female, was advantageous advantageous to the species. And that would follow from natural selection. But now I see, this is in the second edition of his book, but now I see that the whole problem is so intricate that it is safer to leave its solution to the future. I'm afraid that we are in the future, but we don't have solution to it yet. So, yeah, with this I'd like to thank you all.
We have some time for questions. We're also going to have a pizza lunch following, so you can.
Yeah.
So, having had a student doing a thesis on this, but with bryozoa.
Bryozoa.
Yes. The
Yep.
first thing that comes to my mind is how far do sperm travel for fungi? Because the way you're setting up this experiment, you're traveling centimeters. Can they travel meters?
Well, it's a local phenomenon. It depends, of course, on currents. It depends on the local oceanography. But they can travel a long way. I'm sure they can. We haven't done that. We haven't followed particular sperm, but they can travel a long way.
Because
I believe so.
There aren't many bryozoa that do this, but they are mostly simultaneously hermaphrodites. But there's one that has enormous differences in male-female function. And it's very different depending on population density.
Right.
And
Okay.
so
Sure.
Here's my alternative hypothesis for you.
Okay.
And that is that, I don't know about all the other fungids, but the one you worked on where the large individuals are senescing,
Yes.
then they're mostly having male function.
That's right.
That the largest individuals are in fact flooding the environment with sperm, and that would make it smart to be female when you're small.
That is okay, except that the largest individuals, which are male, are very, very poor in reproduction. They're senescent. They're old, and the amount of sperm that they produce is minimal. So in spreading this all over the reef, it really won't work much.
But you're saying minimal per
individual.
Per individual.
That's right.
But most of the individuals that are large.
Most of the individuals are not large, are not getting to that. Well, as a matter of fact, you find that curve also in terms of size groups. Several of them are dying throughout life. Very few remain very, very old. The biggest that we got was an individual of about that size, an old female, by the way, but this was another species, which was 7 kilos. And she produced 2 or 3 eggs, spirit of nothing, but still a female. Yeah.
But I guess what I'm trying to say is that the general environment on the scale of, let's say, 10 to the square meters might be determining reproductive investment as much as
for sure.
what's happening in one spot.
For sure, I agree.
Celoporella, that's exactly
for sure,
what's
for sure.
happening. I'll get you the reference.
Great, thanks.
Any other?
A question about this biological clock, the timing
Right.
of spawning. In the lab, did these animals have natural light?
Okay.
Were there any external things?
This biological clock, as I said, is amazing. No matter if you're in the laboratory, no matter if you're with high light or low light, like clockwise, they will reproduce both in the water, in the field, and in the laboratory exactly at the same time. So, and as I said, they are so accurate in their timing, also year after year after year. So I know that somebody is working on biological clocks, and that's the thing to do in this particular species because they're so accurate. Yeah.
I'm wondering about, it's sort of related to what Jeff was asking. Is there a critical distance? You pair these up right next to one another. Is there a critical spacing that where this kind of phenomenon begins to happen and then drops off?
Yeah, it's a good question. Although in the reef, they're all randomly spread all over, so there is nothing like what I've been doing in putting them far away from each other. So in a way, this is artificial. But doing that, I wanted to avoid any influence of nearby other corals. Now, if there is a particular space or so, I really don't know. I could only guess that if they're 3 meters apart, it'll be okay. In other words, if a female produces some hormones, the male next to her will realize it's a female much more than 3 meters away or so. So,
Big or small
yeah. So I don't have an answer really for that.
One more question?
Regarding the reproductive senescence,
Right.
was that associated with other physiological senescence, other signs of problem, or preceded by other problems, or really this is the only thing you see?
Yes, it's a good question. We haven't done any really other signs of senescence in the soil, only reproductive senescence. So I don't know.
The telomere thing is pretty.
Well, the telomere is the start, except that we haven't really proved it, but at that stage. But it'll be coming. Yeah.
Whether energetically, say, zooxanthella's abundance or whatever?
No, not. I don't think there are. We haven't counted, but I believe that there will be not much change in those antennae abundance or so. But yeah,
All right.
yeah, that's something to look at.
All right. Let's thank Dr. Loya again.
Okay, thank you.
Then we are going to be in the Akubo room immediately following, so feel free to come and eat some pizza and ask some more questions.
I'm so glad to see you. Great. Oh, thank you. Thanks. Well, you know, it has been some years.
Well, yes, I know. Yes.
So how are you? Yeah. All right. Thank you. Thank you very much. Thank you.
Well, I mean, I think you could say you could do the project now in a year if you wanted to, or 4 years, if you wanted this semester. Like, we need to be here in more years. I mean, that's what you should try to do.
Well,
Or just, no,
They're accumulating damage. To Joe and I, we're talking about helping yes, you know, it's getting a little busy.